One of the specialties of the innate immune system is of the first line of defense, preventing microbes from entering the body. The skin is one of the most important of these. Very few microbes can penetrate intact skin. The skin can be broken, however, allowing for penetration by microbes. This can occur by accidental cuts or abrasions, insect bites, injections with hypodermic needles, or even intentional rupture of the skin by microbes themselves, in the case of open sores or lesions. Another instance where we see the innate defense mechanisms in action preventing microbial infection is the acidity of the stomach. The pH of 2 that's found in stomach acid is highly detrimental to most microbes. Mucous membranes line the internal body cavities and also act as internal barriers. The airways in certain areas of the respiratory system have cells with hair-like projections called cilia. These tiny little hair-like projections from the cells are active movers and can push or sweep microbes from deep in the respiratory system up and out into the pharynx. From the pharynx, the microbes are then swallowed and killed by the stomach acid. Smokers are more prone to respiratory infections than non-smokers because smoke damages this system. The enzyme lysozyme destroys cell walls of some bacteria. This enzyme is found in many bodily fluids, including saliva, tears, and sweat. It's important to remember that we also have our normal microbiota. These are bacteria that colonize the skin and mucosal surfaces, especially in our digestive tracts. Any potential pathogen trying to cause an infection has to compete with our normal microbiota. This is their home, and the normal microbiota are not going to give up so easily. The mucosal surfaces of the gut are a particularly good habitat for these little microbes. And with a symbiotic relationship with us, they don't want to leave. Before birth, we don't have any of these microbes, and the immune system is also relatively underdeveloped. This makes infants particularly susceptible to infectious disease. However, after birth, newborns acquire their normal microbiota from breastfeeding and putting other components into their mouths, and especially it's acquired with the introduction of whole food. At this point, their immune system becomes stronger, and they're much more likely to be able to fend off infections. In fact, we're learning that aspects of our overall health are affected by the composition of our normal microbiota. And our normal microbiota is influenced by diet. Specifically, eating more fiber is healthier for our intestinal bacteria, but also our lifestyle, occupation, stage of life, and drug use, especially antibiotic use. Another function of the innate defense mechanism is phagocytosis. Phagocytes include monocytes, macrophages, and neutrophils. These engulf and destroy foreign substances, including bacteria, viruses, and other cellular components. Inflammation is another component of the innate defense mechanisms. Inflammation is when an injury occurs. This could be a paper cut, a wound, or an abrasion, and it results in a series of steps, which include redness, or rubor, heat, calor, swelling, known as edema, and pain, known as dolor. Inflammation results in any sort of injury, so microbes don't have to be introduced to the site of injury in order to get the inflammatory response. So a sign of inflammation does not necessarily mean there is a microbe present. However, what will happen is that the resident macrophages that are in the tissue will initiate a response. In addition to killing bacteria they find by phagocytosis, like you see here, they also send chemical signals known as cytokines to initiate inflammation and recruit neutrophils. Neutrophils are also phagocytes that will engulf microbes, just like you see in this picture. These cytokines also activate the complement system and create signals that promote inflammation in the local area. Remember that all of these components we just discussed are characteristics of the innate defense mechanisms. They are effective from the moment you're born until the moment you die, without changing in their strength or ability to respond to an infectious disease much your entire life. Next, we'll talk about the adaptive immune system. Adaptive immunity responds to the presence of foreign molecules that have breached the external and internal innate defense mechanisms. This relies on antigen-antibody specificity. Remember, the antigen comes from the pathogen or the microbe, and the antibody is made by your own body as part of your adaptive immunity. This antigen-antibody specificity is very specific, much like a lock and key. The adaptive immune system is divided into two categories: humoral and cell-mediated. Humoral is the antibody-mediated immunity, and cell-mediated immunity involves our T cells. Most antigens are large protein molecules associated with microbes, tumor cells, allergens, or foods. They engage and sensitize lymphocytes to generate an effective immune response that may produce antibodies specific for that antigen, in which case we develop humoral immunity, or they can increase the proliferation and number of T cells, which are directed against that antigen. That's what's referred to as cell-mediated immunity, or CMI. T lymphocytes are important players in these CMI, or cell-mediated immune responses. T cells are also broken down into multiple categories, known as T cell subsets. Each category of T cell has a specific role and is identified from molecules on its cell surface, known as clusters of differentiation, or CD molecules. The CD4 molecules are associated with helper T cells, and CD8 molecules are associated with cytotoxic or killer T cells. The helper T cells, identified by the CD4 molecules, are crucial in that they coordinate responses by other white blood cells, especially B cells and cytotoxic T cells. The helper T cells also help with the function of other immune cells, such as those of the innate immune response, like the macrophages and dendritic cells. They exert their function by directly contacting other cells and secreting chemical messengers known as cytokines, which activate the target cells. Now, the humoral immunity, which is antibody-mediated, is mediated, as I mentioned, by antibodies, pictured here. These are Y-shaped structures made by B cells in response to antigens. The antibody structure, as you can see, is made from 4 components: 1, 2, 3, and 4. There are 2 smaller components known as light chains and 2 larger components known as heavy chains. The antibody itself binds to antigens made from pathogens. There are 2 antigen binding sites for each type of antibody. So each antibody can bind to 2 antigens at once. They can bind to antigens on bacterial cells, viral particles, they can bind to toxins, and internal cells, including tumor cells and dead cells. The binding of antibodies to antigens have several outcomes. One, they can activate B cells. Helper T cell lymphocytes can send cytokine signals to the activated B cell, so activated B cells can make more of those types of antibodies. This helps generate a strong immune response to combat an infectious disease. These antibodies are produced with identical specificity, and so just the B cells that are activated are the B cells that are needed to fight off that specific disease. Antibodies can be one of 5 different classes or isotypes. The most common of these is IgG. However, the other 4 are known as IgA, IgD, IgE, and IgM. The Ig is immunoglobulin, another term for antibody, and the isotype is the letter that follows. IgG is what you see pictured here. It is the best characterized of the immunoglobulins known. As a result of the antibodies produced, these antibodies can end up forming lots of different functions. They can inhibit viruses, combine different antigens on cell surfaces, restricting movement of microbes through the tissue. They can group together molecules, such as toxins, to keep them from being toxic to the human body. They can bind to other toxins or viruses and neutralize them. They can target microbes for opsonization or phagocytosis. And they can also activate the complement system, which can create holes in the membranes of microbes. Now, how does all of this work? Considering that there are antibodies that are specific to different microbes. This is explained in what's known as the clonal selection theory. According to this theory, a population of B cells are made in the bone marrow. This happens as a fetus. There are B cells then that are generated for virtually every antigen, including cells and molecules in your own body. Each B cell makes its own one type of antibody, but as a whole, the collection of B cells in your body can recognize many different molecules and microbes. If the B cells initiate a response to anything in the body, in a developing fetus, the B cells recognize that that molecule likely belongs to the human body, and they destroy themselves. This is to protect against autoimmune diseases. However, if the B cell is activated by an antigen later in life, like in a child, teenager, or adult, then that antigen is very likely from a foreign invader, and those B cells respond by growing and dividing and making many copies of themselves to attack the foreign invader. That way, the population of B cells that was selected by the antigen is expanded. Antibodies are highly specific for their corresponding antigen. But in an immune response to a microbe, you are going to be making many antibodies derived from many different B cells. And that's because each microbe contains tens of thousands of different types of molecules that B cells can respond to. This is what's referred to as polyclonal antibodies, because they make lots of different antibodies in response to a single microbe. In the next section, we're going to briefly talk about monoclonal antibodies, before we continue our discussion about the adaptive immune response.